Welcome to video 6 in our course G07 on phylogenomics. In this video, we're looking at rooting phylogenetic trees using two major methods. So the first method uses an outgroup. An outgroup is a sequence or a group of sequences that are very genetically distinct at the DNA level, or indeed the amino acid level if you're aligning amino acids compared to the other sequences, such that there's no evidence of any recent G flow between the outgroup and then the in-group. This helps orient the different paths of mutations that may have occurred over time, such as an AT allele that varies in the sampled population, the in-group, to give it a sense of direction as to whether that changes T to A or A to T. If the outgroup allele is T, for example, and that represents the ancestral state, that may indicate that it's changed to A, and vice versa if it was an A in the outgroup. The outgroup needs to be appropriate level, sort of a Goldilocks zone, that it's not extremely distant, because then it may have multiple substitutions at those bigger alleles like T here, but also sufficiently different that it's not recently related and doesn't have any evidence of recent gene flow. So we can root with a known outgroup, and that essentially means to create an unrooted tree, including that outgroup. We find that outgroup node and set the root to be on that branch towards the base of the most proximate internal node. Another method of identifying the root is midpoint rooting. So that's where there's no clear outgroup. That's maybe a more common situation. Essentially, you create an unrooted tree and you look for the longest span across the different branch lengths that are present, the maximum distance that's possible, and choose that midpoint between those two longest branches. So that's kind of our approach. So we can think about these trees representing diversity, but also evolutionary time, their evolutionary origins. Of course, if we're looking at midpoint rooting, where we have a big branch like this, we're assuming that going back to some ancestor in this branch, that the rate of change over time and across sites is comparable to each other. And we know that that's not strictly speaking going to be true. So it's not necessarily straightforward to estimate the root. And indeed, there are many situations in which a root can't be estimated at all, or it's either not relevant or would not necessarily be informative for the analysis of this great variation in mutation rate across different sites and samples. So we can have an example like this, where you have some kangaroo sequences that's the most divergent sample here. It's labeled as the outgroup, compared to the lemurs and humans, and also to the rodent groups down here. So we think that if we collapse that tree together like a phylogram, what happens here? So we set our root to be here. This is the outgroup node. And then for midpoint rooting, if we do that, we'd assess for the longest possible branches here. So that might be going from A mouse here through this long branch. Oops. A mouse here through this long branch. And then it could really be on E, or indeed along the human branch here, or indeed to lemur. Either way, we're going to hit the midpoint around here. So you can see in each case, the midpoint root is different. And what we essentially do is we construct that tree using this as the root instead. So that's two different options. So if we look at this option of outgroup rooting versus midpoint rooting, we can imagine how they'd be different. So if we outgroup root using kangaroo, that's our root there. And then we have that small internal branch, which corresponds to this one here. Then we have that separation between mouse and rat. That's this long branch here, which corresponds to this one here. And then you see the mouse and rat together, corresponding to these branches here. And then we have this branch leading to lemur and human, and their branches there as well. In contrast, if we have midpoint rooting, so our root is there, we have each of these nodes. So that's the top one here, and that's the bottom one here, T and B. We therefore see the bottom one is here at B, and the top one is here at T. So that's where those two branches come from. Then we have the rodents down here again, the human lemur over here. This branch X is here, and then the E branch is the one listed there. So the outgroup based on that was here. So you can see that the external branches for E is slightly longer. For C, it's about the same. For D, it's about the same. For A and B, it's comparable. But note up here, we have a longer route leading to the rodent lineage. whereas down here it's separated because part of the root is for the kangaroo, lemur, human clade. So that means you can see there for these ones here, the branch lengths are slightly different. So you can see that best on this diagram here. So you can see therefore if we have the unrooted tree like this, what would we get? So see there we have an unrooted tree looking at the maximum distance across the whole tree length. So that's going to be going from A all the way along this branch to either C or D, which would be a pretty bad equidistance, and we might have a root around this point here. So if they're midpoint rooted, you can see that we have A and B here, C and D there. This node there leading to C and D, and then this E over here. If we have outgroup routing, what we can see before, like we had before, the same red line means that the branch leading kangaroo is shorter, the branch leading to rodents is longer. And if we know some things about the generation times, for example, the generation length for rodents is much shorter than for kangaroos and then for primates, that might inform which tree model fit is the best. If we look at other examples here, we have this unrooted tree here. So what we do to midpoint root here is go along this branch, which is clearly the longest, along to C, and then consider which of the three branches are the longest. So probably G for gorilla. So we go along here, and then the midpoint there, maybe somewhere along here in this instance, which also corresponds to the most likely outgroup in this instance anyway. So we then use this as the midpoint root. So therefore, if we also use A as the outgroup, we kind of actually get a similar result, although the exact placement of the root might differ slightly. So that would mean that A is the most divergent sample over here. We create a big tree like that, then a small branch here, down to the split between B and C. So we draw that there. We have, I'm going to put B on top. So that's our branch leading to B, the B node here. Then we need some space for C. The branch is a bit short, but anyway, so that leads to a split here. This is our branch C down here. So we go down to here where we have a split between our branch leading to G, our branch leading to D, which again splits again, going to E, and then F. How long is F? So that's what we get. So that's the chimp and human. That's the gorilla, and orangutan is here. And that's our gibbon branch there. So that's a very badly drawn tree, but it's kind of gives you an indication of what it would look like. If we use the outgroup, then we do the same sort of structure of the tree with a orangutan as an output outgroup, and you can try that yourselves. And what you see is that the structure of the tree will change. The topology is the same, but the structure is quite different. There's another example here where we have an unrooted network, where we have a root shown here along the mouse lineage, because the rest are primate linked lineages. So if again we'd have our mouse as the most divergent example here at the start, we could the root here, and then start putting the primates here, starting with the lineage leading to the tarsier and lemur, and then the rest, and so on. So we get to treat looking something like this. So you can see there the mouse and lemur. The bootstrapping for this internal node is quite weak because it's very, very short. It's stronger for all the other ones, except for this one here between human, chimp, pygmy chimps, and chimps, because there's some incomplete lineage sorting at that point. So that gives us an idea of how we can use outgroup routing. Another example here, just a hypothetical tree. We got nodes A, B, C, D, E. And then we can ask if B is the outgroup, where is the root using midpoint routing? So this is our outgroup using outgroup routing, midpoint routing. We could start C and D seem the same, so it doesn't really make a difference. We go along here, and again we go to A and E, or I'm not sure if A maybe A is longer, but then we get the midpoint at around this point. So we choose that as a split between a longer branch for K and an upper branch for K. So this lower one would lead to A and E. This upper one goes to a split for B and then to C, the CD ancestors. So we have a split here going to B, and then another line here going to the ancestors of C and D, like that. So that's our midpoint rooted tree. We can ask then our CD and E a clade, so CD and E over here. So I'll let you think about that yourself. Are B, C, D, and D a clade? So B, C, D, are they a clade? So looking through different tree options is complicated, as mentioned before. And if you think about the number of possible options, then if we're applying this, then going back to our substitution rate modeling and our optimality criteria and the neighbor joining tree construction, this is complicated because if we have bifurcating trees with n samples, the number of trees required for a rooted one is very, very large. So 50 samples corresponds to about 6 by 10 to the 96. If they're unrooted, it's less, but it's still going to be very, very big. So you can see that's going to really increase exponentially, indicating that if we're thinking about true routing options, once you have many options, this is quite a computationally-intensive task. It's not necessarily trivial. So that's why outgroup routing and midpoint routing are rapid approximation methods that really can tell us a lot about a bunch of samples, especially if they're recently collected. So that's the end of this video. Please see the next video, which will cover the molecular clock.